Good morning, everybody. I hope everyone can hear me. Welcome to today's webinar. So at the beginning, I will give the stage to Jacques. He will introduce you to the White Hat Scientific.
Thank you very much, Amra, and good morning, everybody. Thank you very much for attending our White Hat Scientific hosted webinar entitled Innovative Solutions in the RNA Sequencing Workflow. Anna, would you mind just advancing the slide, please? So for those of you that don't know me, my name is Jacques TraPR, and I am the Clinical and Automations Product Manager at White Hat Scientific. Before we start, I just have some housekeeping rules. Everyone will be muted during the course of this webinar. If you do have any questions, please make use of the chat box function that's available. We will have a dedicated question and answer session where we will answer any questions that you may have. The webinar will also be recorded and the link which will be shared with everyone afterwards. Would you mind just advancing again, Anna, please? So today I have great pleasure in welcoming Lexogen to the White Hat Scientific list of reputable suppliers. Lexogen are the RNA experts, and today we welcome Dr. Anna Skucha as Lexogen expert and presenter. Anna is an application scientist at Lexogen, and she will be telling us more about the innovative solutions on the Lexogen product portfolio. Without further ado, Anna, the floor is yours.
Super. Thank you very much, Jacques, for this kind introduction. So let me start from my side. My name is Anna Skucha, and Jacques mentioned I'm an application scientist at Lexogen. This is within the tech support team. And it's my pleasure today to introduce you to the innovative solutions in the RNA-Seq workflow. So first, few words about the company. So Lexogen is a biotech company founded in Vienna, Austria in 2007, and we focus on developing technologies for complete transcriptome sequencing. We also have an office in the US. This branch was founded in 2014 to support sales and technical support activities in the United States. All R&D services and the manufacturing teams are located here in Vienna, Austria, and providing a sustainable local company culture with interdisciplinary multinational team of RNA experts. Lexogen employs a worldwide distributor network to offer our products tech support and services to our customers all over the world. With this webinar, we welcome White Hat Scientific as our official distributor in South Africa. Lexogen offers kits, solutions for every step of the RNA sequencing workflow, and this is from sampling and RNA preparation to NGS library preparation and NGS data analysis. In this webinar, we will highlight some of our most popular products. And if you have any questions about other Lexogen products, you can contact us or our distributor directly anytime. For the solutions that I will present today, we also offer a service. So you could provide us with any of your input material, and we will prepare the libraries, sequence the samples, and provide you with the sequencing results. Lexogen's team is dedicated to offer innovative cutting-edge technologies that support the research community to perform state-of-the-art science. In this webinar, we want to highlight some of our customers' research questions and how Lexogen products help them to publish their results. So in this webinar, we will be focusing on gene expression profiling, whole transcriptome RNA sequencing, small RNA isolation, and small RNA sequencing. We really value your feedback, so we prepared 3 very short questions that will be popping up during this presentation, and I would really appreciate if you took just 10 seconds to answer very simple questions. So the first one is, think about setting up an RNA-Seq experiment. What is your major challenge? Is it planning and setting up the experiment? Is it the handling issue, so you're not confident with preparation of the libraries? Is it about finding a suitable facility to process your samples? Is it data analysis, which is often a bottleneck? Is it handling many samples at the same time, getting a budget, or maybe you have other problems? And yeah, we will be very interested to hear your thoughts on that. Super, that was quick. Okay, so many researchers are looking for the fastest and easiest way to assess differential gene expression to get a quick overview over the changes in expression levels of protein coding genes. The next generation sequencing is the method of choice for such global analysis, and Lexogen QuantSeq library prep kit proved to be the best solution for all those applications. Using QuantSeq, the researchers were able to drive their projects and identify promising compounds, select the best clones, characterize immune responses, or to investigate signaling pathways involved in oncogenesis. And just a quick note from my side that the slides will be shared and you can access each of these publications as well as a webinar indicated by this icon right after the talk. QuantSeq Lexogen's 3' mRNA-Seq library prep kit offers the most efficient differential gene expression solution. You save costs because QuantSeq enables a very high level of sample multiplexing. The protocol is very fast, easy, and very robust. QuantSeq works on a wide range of input material, also on low input, low quality samples, including FFPE RNA. Why do we think that QuantSeq is better? So in the standard mRNA-Seq workflow, multiple fragments are generated per transcript, and you end up with fairly involved bioinformatic analysis. In contrast to the common mRNA-Seq method, QuantSeq generates only one fragment per transcript, located at the very 3' end of polyadenylated transcripts. Consequently, much fewer reads are required per sample to get the meaningful gene expression data. This enables a higher degree of sample multiplexing in one sequencing run. Additionally, the workflow is fast and easy, and the downstream data analysis is particularly straightforward. All these factors combined make QuantSeq a very cost-efficient method for expression profiling. QuantSeq features a very fast and robust protocol. It consists of only 5 major steps and uses total RNA as input. QuantSeq requires no pre-processing steps, and here I'm talking about rRNA depletion or selection of polyadenylated transcripts up front, as required for other workflow. This saves a lot of time and consumables. QuantSeq libraries can be prepared in 4.5 hours with the required hands-on time of 2 hours in total. As mentioned before, QuantSeq generates only one fragment per transcript corresponding to the very 3' end of mRNAs. This is achieved by an oligo-DD' reverse transcription to generate first-strand cDNN. Following this, the original RNA molecules are removed from the samples, and the random second-strand synthesis is carried out to produce a double-stranded cDNA libraries. In both first and the second-strand synthesis, the partial Illumina adapters are included in the 5' end of each oligo. Library generation includes purification steps to remove any small artifacts and primers. The libraries are then amplified by PCR, and the complete Illumina adapters are incorporated for clustering on a flow cell. After amplification and purification steps, your libraries are ready for quality control and the subsequent sequencing step. After showing you how QuantSeq works, I want to highlight one of the before mentioned applications. So in this study, they investigated different responses to hepatitis B vaccine. 34 healthy, non-vaccinated individuals were recruited, and the vaccine was administered on day 0 and day 13. Then the transcription profiling samples were collected at day 0, day 3, and day 7. Hepatitis B antibody titer was checked at day 0, day 60, half a year, and 1 year post-vaccination. So this way, researchers identified a group of non-responders, low responders, and strong responders to hepatitis B vaccination. And while looking at the transcriptome results, it turned out that the responders showed strong differential gene expression early after vaccination, while non-responders present with less significant differential gene expression, which was also delayed in time. Additionally, it became apparent that differences between responders and non-responders are particularly strong at day 0. So taken together, we can conclude that QuantSeq results help to identify individuals predisposed to good reaction to hepatitis B vaccine. Low-quality RNA, including FFPE samples, could be used as input for library generation with QuantSeq. And we have an excellent case study where archived, highly degraded RNA was used for RNA expression profiling. You may be familiar with a DV200 index, and this is the value that represents the percentage of RNA fragments that are longer than 200 nucleotides. So in this webinar, the researchers were able to generate meaningful data from single-digit DV200 samples. So that means that majority of mRNA fragments were shorter than 200 nucleotides, but this did not constitute any limitations for the kit performance. The results, so the number of detected genes is comparable between FFPE and fresh frozen RNA. So this highlights that the maximum information is provided despite the low quality of RNA. I just wanted to mention that we do have a set of protocol modifications for customer working with heavily degraded material. How is it possible that QuantSeq works that well on degraded material? It's depicted here. So as outlined at the beginning, QuantSeq is a 3' mRNA-Seq library prep method. This means that the libraries contain fragments located at the 3' end of transcripts right before the poly-A tail, and only one fragment per transcript is then sequenced. As highlighted on the previous slide, RNA derived from FFPE samples is usually degraded to some extent. However, even if poly-A tail was partially shortened due to sample degradation, the oligo-DT primer is able to bind to the remaining poly-A stretch and generate a functional library. It might be a little bit shorter than it usually is for the intact RNA, but most of the fragments that can still be used for the sequencing. And yeah, the data you receive is of good quality. For this reason, many researchers succeeded to analyze FFPE samples using QuantSeq in several applications, such as library generation from patient samples stored in biobanks, library preparation from patient samples in studies focusing on breast cancer and uterine fibroids. Again, the slides will be shared, so you have access to all these publications as well as webinar indicated by this icon. So far, I have described the use of QuantSeq for everyday applications in the lab with usually relatively small projects with up to several hundreds of samples. In this case, each sample is processed individually and the resulting libraries are QC'd and then pulled for the RNA sequencing run. After sequencing, the samples are identified by unique indices that were introduced during the library preparation. However, many researchers have hundreds or thousands of samples to process in parallel. For these purposes, Lexogen offers two solutions. It's either the automation of the QuantSeq protocol on the liquid handlers or the variation of the QuantSeq protocol called QuantSeq-Pool. So here comes the time for the second pooled question, and we have just few seconds to answer the question, how many samples do you usually process in one RNA-Seq experiment? Is it below 24, so it's like small project? Is it a mid-sized project of 96 samples or more than that? We will be very happy to hear your feedback on that. Thank you. So I see that many of you process 96 samples or more, and we know that automation is a key factor for such high throughput labs, and also this is the case for the biotech industry or sequencing facilities. So the good news is that QuantSeq can be automated on commercially available liquid handlers, and the automation scripts were already validated on a number of instruments. The setup have been mentioned in several publications, and QuantSeq automation has also been discussed in one of the most recent webinars. Another way to approach such high throughput projects is QuantSeq-Pool. We have released QuantSeq-Pool in summer 2020, and it is being already used by a number of customers in high throughput labs. QuantSeq-Pool provides you with an enormous degree of sample multiplexing, and manual processing of nearly 37,000 of samples has never been that straightforward. You save up to 90% of lab consumables and sequencing costs, and I will tell you how that happens. So how does it work? QuantSeq-Pool follows the same principle as QuantSeq. You start with reverse transcription from the poly-A tails, then you remove the RNA, you perform the second strand synthesis and the PCR. However, in contrast to the standard QuantSeq, the samples are not processed individually. Up to 96 samples are uniquely labeled at the very first step during the reverse transcription. Then they are pulled into a single reaction and the downstream processing happens in only one tube. What are the benefits of pooling? Up to 96 samples can be processed in one reaction. This saves a lot of time because the libraries do not need to be QC'd and normalized individually. Processing one sample pool versus 96 individual preps saves a significant amount of plastic wear. And this way you also minimize the technical variability. So as mentioned, the multiplexing capability is unbeatable. Using QuantSeq-Pool, you can multiplex nearly 37,000 of samples. So far we covered QuantSeq and the alternative protocols, but the sequencing of the 3' end of polyadenylated transcript is a recommended technology for the expression profiling only. So what if your research focuses on, for instance, isoform discovery, alternative splicing, or SNP detection? These projects require transcriptome analysis methods. And I will shortly introduce you to our solution for the whole transcriptome analysis. Before that, we have another question in the pool we would really appreciate your feedback on. So this time we ask you, what is the key decision maker when choosing a library preparation kit? Is it the time it takes to have the libraries ready? Is it the quality? So do you want the best results? Is it the publications that are available for the preparation kit? Is it the automation? Maybe that's the price, or maybe there's also some other factors that you could indicate here. Thank you very much for participating. Lexogen's solution for the whole transcriptome analysis is called CORALL. So CORALL works with any input RNA, including low input and degraded samples, and offers all-in-one solution with built-in unique molecular indices. The libraries are fully compatible with Illumina sequencing instruments for single read and paired end sequencing. Moreover, the protocol is compatible with automation. The performance of CORALL was internally benchmarked and the kit was compared with two conventional competitors. So here we can see that CORALL delivers an excellent gene expression rates matching competitor kits over a wide range of input amounts. And here we can appreciate that 95% of genes were commonly detected between CORALL and other two competitor kits. So far, we have addressed problems where researchers were interested in getting a fast and efficient overview over a gene expression patterns. But when it comes to validation of drug candidates or detailed analysis of patient samples, or maybe when in-depth transcriptome analysis is required, we need a whole transcriptome NGS approach. The scientists do not only want to count the genes, but they want to see the full length of each transcript to also get the information on isoforms. For these purposes, they use our whole transcriptome library prep kit CORALL. In these selected applications, CORALL was used in combination with poly-A RNA selection kits, so only the protein coding mRNAs were sequenced in full length. Selecting all protein coding genes via mRNA selection will restrict the analysis to poly-A containing transcripts. Many researchers aim at even more neutral approach. They want to analyze all long RNAs, no matter if they are polyadenylated or not. For these purposes, we also offer RiboCop ribosomal RNA depletion kit to be combined with CORALL, the whole transcriptome kit. This way, only the undesired RNAs, rRNAs, which makes up to 98% of each transcripts in your samples, will be removed from the analysis. CORALL was released in 2019, and already a number of publications using this method are available. So a few words about the RiboCop. RiboCop is our product for our RNA depletion, and it works on a wide range of input materials. So that's from 1 nanogram to 1 microgram. The method utilizes magnetic bead workflow and usually and uses a highly specific set of probes. We currently offer specific protocols for rRNA depletion from human, mouse, rat, and bacterial samples. A version of the kit depleting rRNA and globins from blood samples is also available. RiboCop is a probe-based method and works via hybridization of rRNA affinity probes followed by a magnetic bead purification. Lexogen has optimized an algorithm for the state-of-the-art probe design for the best depletion efficiency and minimal off-target effects. The depletion protocol takes around 90 minutes and can be automated on available liquid handlers. Our RiboCop kit shows an excellent depletion efficiency across the species, down to 0.1% of remaining rRNA after depletion. This outperforms all parameters that are advertised by other rRNA depletion kits available on the market. For example, Illumina claims depletion efficiency down to 6% of remaining rRNA from human samples with their new RiboZero kit, but also NEB and QIAGEN, they show higher levels of remaining rRNA according to the available marketing materials. As mentioned before, Lexogen offers a RiboCop version for depletion of bacterial ribosomal RNA. For bacteria, we have 3 different solutions. So the first one is G- for gram-negative bacterial species, such as E. coli, G+ for gram-positive species, such as Bacillus, and the Meta kit for depletion from mixed samples, like microbiome or environmental samples. The Meta kit can also be used as all-in-one solution to deplete any bacterial samples, even if you work with more exotic species. The performance of the Meta kit is depicted here as an example across several gram-negative and gram-positive species. Depending on the species and the amount of input that you use in your experiment, you can deplete down to 0.1% of remaining rRNA. Many researchers want to go beyond the routine analysis of the transcriptome. They want to understand how the changes in transcript levels have been achieved. And gene expression is heavily regulated by small RNAs, such as miRNA, siRNA, snRNA, or pVRNA. There are also other RNA species. Thus, global analysis of small RNA levels give insights into regulation of expression profiles, and small RNA sequencing is a hypothesis-free way to approach the small RNA analysis. For an efficient small RNA sequencing, it is highly beneficial to adjust the method of RNA extraction to your needs. Ideally, you use an RNA extraction that is specific for the small RNA of interest. There are many known methods to extract or select small RNAs for NGS experiments, and they are depicted here. You can choose, for instance, a column-based small RNA extraction kits. And these are easy to use, but they are not very specific, since they apply a general cutoff of around 200 nucleotides and simply elute all RNAs that are smaller than that. This way, you will get all small RNA fragments, no matter if this is a small RNA or just a degraded RNA fragments. To improve the specificity, you can use gel purification and define the size range of the RNA of interest. This is, however, very tedious. It takes a lot of time, and you also lose a lot of material. Also, this way, you still have a small degradation products included in your fraction, and you won't know the difference between the real small RNA or these degraded fragments. Researchers that know exactly which small RNA they are looking for, they can also use the co-immunoprecipitation of small RNAs with their respective agoprotein. And the agoproteins are the proteins that each small RNA interacts with when it functions in the gene expression regulation. This approach is possible only if you have access to antibodies targeting agoproteins and you know exactly which ago interacts in the tissue or species that you work with. So this approach is not globally applicable since you are also limited to the available antibodies. TraPR is our Lexogen solution that combines all the benefits of here named methods. It is fast and easy column-based method, but only isolates small RNAs that are bound to agoproteins, but any agoproteins of any species that you use. So this makes TraPR universal and specific at the same time. TraPR stands for trans-kingdom rapid, affordable purification of risks. It is a column-based approach, and it's a very simple method. You simply lyse your cells or tissues with the provided TraPR lysis buffer. So here you end up with the lysate of free RNA, DNA, proteins, and protein nucleic acid complexes, including the native risks complexes. The lysates are then passed through the ready-to-use TraPR columns, and the column worked by ion exchange chromatography, and it retains all free RNA, DNA, while the native risks passes through the column and are eluted in the elution fraction. The elution fractions can be then used for extraction, and the extracted fraction is then accessible for any library preparation kit. TraPR is universally applicable. It is faster than any comparable method introduced before, and it's affordable. You only need a column per prep. You do not require any specialized equipment, such as large gel apparatus required for gel extractions, and a simple benchtop centrifuge is all that you need. The purification procedure takes approximately 15 minutes, and the workflow is compatible with different input sources, such as plasma samples. We have released TraPR, the functional small RNA isolation kit, in summer 2020, and this new method is already used in some publications. In combination with Lexogen's small RNA sequencing prep kit, TraPR enables most efficient sequencing of physiologically active small RNAs. So this was just a snapshot of how Lexogen portfolio can support researchers around the world with innovative, high-quality RNA analysis solutions. You can see that we have only discussed QuantSeq for gene expression profiling, CORALL for the whole transcriptome analysis, RiboCop for rRNA depletion, and TraPR for functional small RNA selection. We offer many more products within the RNA-Seq pipeline. This is from metabolic RNA labeling and data analysis solutions to NGS controls. I would just like to encourage you to explore our website for more information. However, what if you have a limited time or limited expertise to establish RNA sequencing experiments in your lab? We can offer all our products as a service in our service facility here in Vienna, Austria. You only have to ship us your sample, whether this is a frozen cell pellet or maybe already extracted RNA, and we run the entire process, including data analysis for you. Our service facility offers personalized treatment with individual consultation and customer-tailored approach, peace of mind with our project management and technical expertise, and high-quality results thanks to our competence to handle samples even outside of the standard kit specifications. With this, I would like to thank you very much for your attention, and we can move to the Q&A session where you can ask us anything you're still interested in.
Thank you very much, Anna, for that very informative and interesting presentation about all the innovative solutions. I see we have two questions that has come up in the chat box. So number one, about TraPR. Does TraPR directly extract RNA, or does it need to be coupled with an extraction kit?
So the extraction procedure is included in the TraPR user guide. So you have to follow the user guide's recommendations, and then the last section of the protocol will guide you through the extraction of small RNAs from the risks complexes eluted from the column.
So if we understand correctly, it is an all-inclusive.
Yeah, that's correct. Some of the reagents need to be provided by the customer, but most of them, including the columns, the most important part of the kit, are included in the kit. Mm-hmm.
Right. So second question about CORALL. Is CORALL compatible with FFPE samples, and what is the recommended input amount?
Yes, CORALL is compatible not only with degraded samples, but we also have an excellent data sets from our customers showing that CORALL is compatible with FFPE samples. So there are very minor protocol modifications when the customer is handling FFPE samples, and they are collected in the user guide. So the customer can easily find them at the end of the protocol of the booklet. Regarding the input amount, so the CORALL can handle 1 nanogram to 1 microgram of total RNA, and this is the input for before the pre-processing, such as rRNA depletion or poly-A selection. And if a customer is working with total RNA and doesn't want to include any pre-processing before the library preparation, the input to work with is between 1 nanogram and 100 nanogram.
Excellent. Next question regarding RiboCop. Can RiboCop be used to deplete ribosomal RNA from degraded RNA samples?
Yeah, that's correct. In fact, this is a preferred solution if a customer is working with degraded samples. So if you can choose between rRNA depletion and poly-A selection, while working with degraded material, rRNA selection is a better approach. So RiboCop has been tested on degraded samples, including FFPE RNA, and can be definitely used for this application.
Okay, and then we have another one. What is the length of cDNA generated by random priming? I'm not sure which kit this is referring to.
So I assume that this will be, actually the random priming would be included in both QuantSeq and CORALL. It might be difficult to explain that without the respective workflow. But so just briefly, the first steps of QuantSeq. This will start with the oligo-DT primed reverse transcription. So we have an oligo-DT primer binding to the poly-A tail of mRNA, and this extends the first strand synthesis. In the next step, there will be a random priming occurring on the first strand cDNA. But only the very free prime end fragment will be extended and will give rise to the libraries afterwards. So the final library, in order to be efficiently amplified, it requires Illumina adapters and only the very free prime fragment. So only this random primed fragment generated at the very free prime end of the polyadenylated transcripts will be amplified in the last steps of the protocol. I would be happy to elaborate on that if the customer needs further information.
I think if anybody has additional questions, we have a few more that we can answer, but if anybody has any questions, please feel free to reach out to us afterwards and we can address those. Then another one, what kit or approach would you recommend to obtain both transcriptomic profile and SNPs?
So the SNPs, for SNPs, I will start from this part of the question. We would probably recommend CORALL, as you cover the whole transcript versus only the free prime end is covered by QuantSeq. So CORALL is known as more standard approach for RNA-Seq analysis, and this would be applicable for this application.
Excellent. Then another one regarding RiboCop. It's also a question that I had initially. So is RiboCop being developed for plants, and does the current RiboCop work on plant or ribosomal RNA?
So the current RiboCop contains probes that were designed specifically against human, mouse, rats, and bacterial samples, so bacterial rRNA. So the plant species are not covered. Depends on the application. We usually encourage our customers to check whether our kits would work for the species they work with. They may observe a minor depletion of rRNA, which is not always best for NGS applications, but it might be helpful. In case there is interest for other species, we are happy to hear about that, as we could address these needs in the future. I just have a colleague here who mentioned that the plant kit might be in development. So stay in touch.
Great to hear. Yeah, and I don't see any other questions. I think we'll leave it at that. As I said, thank you very much for a great presentation. To everybody that attended, thank you very much for taking the time to listen to the solutions that Lexogen has on the portfolio. We're very excited to welcome Lexogen to the Whitehead Scientific family. And as I mentioned, if you do have any questions or interest in quotes for respective products, please feel free to reach out to the Whitehead Scientific team. Yeah, with that, thank you very much. We'll leave it at that and enjoy the rest of the day.
Thank you for your interest, and hope to see you soon or hear from you.
Thanks, everybody.